The polymerase chain reaction is a technique which allows one to amplify a specific region of the genome more than 1 billion-fold. A reaction is set up which contains a small amount of genomic DNA, 2 oligonucleotide primers which hybridize to either side of the region to be amplified, nucleoside triphosphates, the substrates for DNA synthesis, and the heat-resistant DNA polymerase, Taq polymerase. The reaction is placed in a PCR machine, which controls the temperature of the reaction. Amongst the many DNA sequences present in the tube, only the DNA located between the sequences that are complementary to the primers is amplified. This is referred to as the target DNA. The first step in the reaction involves denaturation of the genomic DNA by heating to 94 degrees centigrade. The reaction is then cooled to 42 degrees centigrade, allowing the primers to hybridize to the complementary sequences in the genomic DNA. The temperature is then increased to 72, the optimal temperature for Taq polymerase to synthesize DNA, and this completes the first cycle. Cycle 2 begins with the denaturation step, followed by the hybridization of the oligonucleotide primers, and then DNA synthesis by Taq polymerase. At the end of cycle 3, 2 of the products contain only the region between the oligonucleotide primers. At the end of cycle 4, 8 target molecules have been generated and 8 longer length molecules. The number of target sequences continues to grow exponentially until by cycle 30 over 1 billion target molecules and only 60 longer length molecules have been generated. The loading dye is then added to the reaction and the DNA fragments are separated by electrophoresis through an agarose gel. The amplified target DNA can then be eluted from the agarose gel for further analysis.